Hi, good afternoon, everyone. My name is Mel, and I work in Adrian Whitty's lab, which is a biochemistry lab. So today I'll be presenting to you what I did this year. So my project is called Using Peptide Synthesis in Flow and Click Chemistry to Create Small Bivalent Activators of the RET Receptor Tyrosine Kinase. So there's a whole lot of jargon in my title, so please don't worry about it. We'll go over it in detail in a bit. But first of all, so since I work in a biochemistry lab, all of our science and all of our research is framed within a specific biological context or biological frame. So we're first going to begin by looking at the biological system that I worked with. So in my research, I looked at the RET signaling cascade, which stands for rearranged during transfection. And there's mainly two things that I think are important to recognize from this image. So the first thing is that RET, which is shown here in dark blue, is both outside of the cell and inside of the cell. So therefore, that could mean that things outside of the cell can bind to RET and then affect change inside the cell. And this change is seen via phosphorylation, which is shown right here in these red circles with the P's. And so phosphorylation is sort of the currency of change within the cell. Additionally, another thing to note about this particular signaling cascade is that two things need to bind to RET in order to enact this change. So GDNF, which is a ligand, so a ligand is anything that will bind to a receptor, needs to bind to a co-receptor, so RET is just special and needs two receptors because it has issues. So we need both the complex of the ligand and the co-receptor to bind to the receptor to enact change. So now we're going to just look into why I specifically care about this very needy receptor. So I'm particularly interested in this particular cascade because GDNF, which is the ligand, the little light orange ligand that I showed on the previous slide, has been shown in animal models of Parkinson's disease to actually promote neuronal survival. So GDNF has actually been shown to protect neurons that would otherwise be degraded by Parkinson's disease. So therefore, this makes GDNF a molecule of interest because it has this possible therapeutic benefit. But the issue is that GDNF is a pretty big protein. And so a protein is just a big complex of amino acids. Typically, it's about 384. So because GDNF is such a big protein, it's unable to cross the blood-brain barrier, so it can't get into your brain. So if you wanted to see these therapeutic effects of GDNF, you'd actually need surgery, which is unpleasant, and like surgery within your brain. So definitely not the best drug delivery method. So therefore, we have sort of this leads to the question is there actually a molecule that can act like GDNF and promote this type of therapeutic, promote these therapeutic, benefits, but that's small enough to actually pass through the blood-brain barrier. So then this leads to the goal of my project, which is actually not to develop a small molecule agonist. So an agonist is, again, anything that just promotes survival of a particular cell. So the purpose of my project isn't to actually create a small molecule agonist that can pass the blood-brain barrier, but just to create a model or a prototype of one with the, to act as a guide for future development of a small molecule. So in developing this prototype, what we're going to do is trying to recognize and account for the key aspects of GDNF that we think are important to model and to create a small molecule that would actually be capable of crossing the blood-brain barrier and allowing for these therapeutic benefits. So, and one particular aspect of GDNF that we'd like to copy in our prototype is this bivalent nature of GDNF. So I haven't discussed this at all, so it's okay if you have no idea what that means. So basically, GDNF is essentially two molecules that are exactly the same and that are stuck together. So they look like two blobs. And essentially, GDNF needs to exist in this form in order to promote change inside the cell. So if we were to look at this in a cartoon form, what this would look like is we have GDNF, our original protein, again in that homodimerical form. And then what I'm going to do for my prototype is take GDNF and create a peptide of it as a small molecule. So a peptide is just a portion of a protein. So while a protein is about 384 amino acids, a peptide is like 20. And I'm going to scaffold them together so we get that homodimerical form. And then what I hope to see in cells is that my prototype will then bind to my co-receptor, and then they will come together and then bind to RET, and then we'll achieve activation, which would eventually lead to our therapeutic benefits. All right, so next slide. So this is a general overview of the workflow of my project. But before we get into how I actually did it, it's important to sort of look at how I designed the specific prototype that I used. So this is an image of GDNF and GFR1 interacting. So GDNF is shown in dark blue. And as you can see, if we were to cut this molecule in half, they're mirror images of each other, which indicates, again, that it's a homodimer. And again, this is something that we tried to mimic in our prototype. Additionally, the next thing that is important to see is that GDNF interacts with GFR alpha 1, which is our co-receptor, again, because RET is a needy receptor, through this one particular part of GDNF. And you can see it's kind of in a cyclic form. And again, we tried to mimic this type of cyclization within the interaction. So now if we take a close-up within this circle, what we see is, so this is our close-up, and here we have, again, GDNF, different coloring, and this region is called finger 2. I didn't name it, it's just simply what it's called. And here's GFR alpha 1 in gray. And based off of the literature, we were able to identify the key contact amino acids that actually promote binding and allow for binding. So for GDNF to actually bind to GFR alpha 1, and those are shown in purple. So they're here. These are our amino acids, and down here in the actual sequence. And then our wheat colored residues are non-contact residues, so they're not important for binding. And since you can see they're on the ends, we just got rid of them because we didn't want to bother having to synthesize them. So we're like, no, thank you. And then these red residues are also non-contact residues, but because they're interdispersed between our contact residues, we needed to include them in our peptide design. So both the red and purple residues resulted in what we actually synthesized as our peptide. So now we're going to get into how this was actually done. So this is more based off of the chemistry. So there's only two sort of cool things to notice. So we synthesized our peptides using FMOC, solid support peptide synthesis. And basically what that is you're synthesizing your peptide on resin, which is pretty cool. So it's like a little polymer, and then you just add each amino acid sequentially, and it was a lot of fun. And then additionally, we use flow chemistry, which is a really cool method because it's much faster than traditional chemistry methods. And it also increases the efficiency a lot because everything is in flow. So all of your reagents are all flowing through your reactor, which is pretty awesome. All right. And now we're going to get to how we cyclize the peptides because, again, we wanted to mimic this finger form and this cyclic form as I showed earlier. So again, here is our image of that. And we found two non-contact residues shown in blue, and again shown in blue. And we mutated them in our sequence so we could form a linkage between them using this reaction. So we get a nice circle. And then finally, we attached the peptide to the scaffold to form that homodimer. And this was done using click chemistry. So in the end, we get a nice triazole ring. And you can see that ring right here. So this allowed our linker to be actually to attach to our peptide. So this is the final product here, our small molecule prototype. And we can see that it's symmetrical and also cyclic. And again, here we tried to copy the fact that it's a homodimer and then it's cyclic right there. And then lastly, so the next steps of this project, which I guess obviously I won't be doing, is to actually be able to test these small molecule prototypes in cells to see if we can actually get activation. And I'd lastly like to thank the wonderful Witty Lab who has put up with me for a very, very long time. So with that, I am all done. Thank you so much. Yes, Tyler.
For your for your original findings, the literature that you discovered said that GDNF helps with the neuronal growth. You said it can't cross the blood-brain barrier, but how do they discover that it has this neuronal growth? Is it like, do you know what I'm trying to say?
So this particular pathway has been shown to sort of be involved in neuronal survival. So and it was sort of postulated as a possible good.
So it was never tested for its actual
Um,
ability?
It's possible. I personally haven't read the literature on that. Yes, Dr. Gilmore.
So have people shown that GFR alpha, if you just dimerize it, that's sufficient to activate the
They
pathway?
haven't. So this actually makes this project very high risk in the terms of that we know that bringing together two RETs achieves activation, but the bringing together two GFR1s has not been shown to be sufficient to achieve activation, and partially it's because we don't know the actual mechanism for how RET is activated. For like GDNF in complex with GFR alpha 1 is just sort of the most commonly like acceptable way to look at it, but then there's been mutagenesis data showing that if you make GDNF incapable of binding, RET is still activated, which would suggest that GFR1 and RET are in complex as well. Yes, Dr. Dorr.
One of your last few slides, you have this very nice detailed molecular structure of the whole cyclic entity that you created with triazole.
Yes.
So that diphenyl unit with the four-membered cyclic ring jumps out at me. I'm sorry if I missed why that's there.
Right here?
Yes.
My project was done in collaboration with the Buehler Lab. So this is a delta trioxanate that I think the Buehler Lab synthesized in 2014. And the idea was that it's pretty sterically hindered. As you can see, it's a tight ring.
Mm-hmm.
And using this particular scaffold while making while increasing steric hindrance and making the formation of this product more difficult increases the novelty. And also we were hoping that by using this particular scaffold that we would achieve the rigidity that we think we need in order to achieve possible activation.
Okay, cool. Thank you.
You're welcome. Definitely though, to create like a more a peg linker should also be tested just in terms of comparison, because
Those are very, very, very different functionally.
they are, yes,
Okay.
most definitely.
Thank you.
All right.